chair of the Energy and Climate Committee in the Sierra Club San Francisco Bay chapter. I'm here with Susan Stepanek, also a member of the committee, who will help facilitate the meeting this evening. We're both happy that you were all able to come this evening to hear our guest, Dr. Danielle Stevenson. Before we get started, let's do a quick review of our agenda and meeting guidelines. So after I introduce our guest, we'll give Dr. Stevenson a good 50 minutes or so to present, followed at 8 o'clock or thereabouts for questions and discussion. For now, if you'd like to introduce yourself, please put your name, location, and any affiliation in the chat. During her presentation, please remember to keep yourself muted, and you may optionally add questions to the chat. And after the presentation is finished, if you have a question, please raise your hand to ask it and wait to be called upon. Any questions? Okay.
Great.
And we are recording, correct, Susan?
Yes.
Okay.
And
To set some context for the presentation this evening, nature-based remediation of contaminated land. A while back, Jean Wu, a member of the San Francisco Bay Chapter's Energy and Climate Committee, heard our guest speak in another venue. She was so impressed that she suggested that we invite Dr. Stevenson to present to us on how fungi and plants can be applied to decontaminating industrial sites. This capability should be of particular interest to the Bay Area, given the large number of toxic sites we have, many of which will spread contaminants as sea and groundwater levels rise. Plus, the recent announcement that the Valero oil refinery in Benicia is slated to close next year calls attention to massive sites of contaminated soil at the Bay Area's refineries, all eventually destined to close. As plans are made for remediation, it is important that communities have the opportunity to learn about how working with microbes, plants, and fungi together can initiate the process of regenerating a healthy and biodiverse ecosystem. Dr. Stevenson will describe what is possible beyond leaving contaminated soil capped in place or digging and dumping it elsewhere. Danielle Stevenson is founder and president of the Center for Applied Ecological Remediation. She is a multidisciplinary scientist, mycologist, and environmental problem solver who works with soil, fungi, plants, and people to address waste and pollution in creative and circular ways. She holds a Bachelor's of Humanities from the University of Victoria and a PhD in environmental toxicology from the University of California, Riverside. Her dissertation research focused on bioremediation of brownfields with fungi and plants. She also founded and runs DIY Fungi, established in 2012 for research, education, and action around fungal food, medicine, waste management, and remediation. And Healing City Soils, established 2015 with the Compost Education Center to provide soil metal testing, resources, and community bioremediation for people growing food. She currently serves on the Department of Toxic Substances Control's Equitable Community Revitalization Grant Treatment Technology Council and the Board of Co-Renewal. She is involved in many projects and organizations around the world supporting regeneration of lands and waters, environmental education, and community capacity building. Dr. Stevenson, Please tell us more about what you found.
Thanks, Rebecca. I'm just checking you can see my slides.
Yes.
Yay. Okay, great. Thank you for having me. I'm excited for this discussion. So I have targeted a bit towards the Bay Area and oil refinery or oil field contamination. But I'll also just give an overview of this problem and the potential of working with fungi and plants or biological options for remediation and give some case studies, mostly from my own research and efforts. So I just always like to start a discussion or presentation like this, but this focus on remediation, so addressing pollution in the environment by talking about where I'm from and how I got into this type of field. So I grew up in a really polluted place in Lake Erie, and anyways, there's, everything from oil spills in the lake, the tributaries to which used to catch on fire from the oil. There's, toxic algal blooms from industrial agricultural runoff. I know that there's similar issues in the Bay Area as well. There's the second largest floating trash island in the world. There's nuclear plants that run their cooling water into the lake. There's just so many issues in the area stemming from the industrial activity there. And it's so bad that it's actually mentioned in the original version of the Lorax. There's this line I highlighted: I hear things are just as bad up in Lake Erie. And um, the reason I start with this is because this line actually was taken out of the Lorax. If you read the Lorax today, it won't say this, and that's because I guess people sort of petitioned to have it taken out, claiming that the lake has been substantially cleaned up since the 70s or so. So and it has there's still a lot of pollution issues there, but it has markedly improved, and it's not because of remediation efforts. So it's not because of efforts to clean up the pollution. It's because of efforts to stop the pollution from entering the lake in the first place. So I like to sort of ground a conversation, and I feel I sense I'm probably among like-minded people here, the Sierra Club, just by saying, by working on and advocating for bioremediation to occur to clean up existing pollution. I'm in no way advocating for pollution to continue. So I think that the most successful approach will be one that actually stops sources of ongoing pollution from occurring and cleans up legacy pollution that we're already dealing with that's already harming our ecosystems and communities. So, this is a really big problem. At least 63% of the world, so of the land around the world, is affected by soil pollution. This is a statistic from the United Nations Environmental Program from about 4 years ago. And that pollution is impacting us in ways that could be a whole presentation on its own. But for now, I'll just keep zooming in. So, across the United States, there's a very conservative estimate of at least 1 million contaminated sites across the country shown in this map, covering more than 200 million acres. These are just sites that are regulated by the Environmental Protection Agency. So there's a huge number of sites that are not represented on this list. But even still, you can see that the entire country is is very polluted, with different types of contaminated sites. Just some terminology. as we move forward. You might have heard of Superfund sites. Um, these can be larger and more highly contaminated, but it's not tied to the definition. They're defined through being part of a program that was established to address abandoned hazardous waste sites by suing the responsible industry. And that those funds go into a super fund, which funds the cleanup of these really contaminated sites. And then we have brownfields, which are just properties. They tend to be urban properties uh, that are basically underutilized because there's contamination or there might be contamination there. There's a lot of other types of contaminated sites too, but um, we'll just focus on those for now. So now we'll zoom into California. We actually have the second highest number of contaminated sites after New Jersey. Um, so there's at least 200,000 brownfields, just that one type of contaminated site, um, shown in brown on this map I made in the state. And I did some spatial calculations to determine, again, major underestimate, but at least just the contaminated sites regulated by the Environmental Protection Agency cover about a tenth of the landmass in this state. I'm going to zoom in on LA County for now, but this trend is evident in the Bay Area absolutely as well. I haven't made a map about it, but I have made a map about LA County. So this map is showing the essentially concentration of these contaminated sites within communities of color. So in this map here, an area that is shown in white is an area with a high percentage of white folks. And you can just see that the contaminated sites in LA County are just pretty much entirely concentrated within Black and brown communities and low income communities. And so now I pulled up a map of, current Bay Area contaminated sites. I put the link. You can check this out. So this, again, there's like the different colors and shapes are showing just different types of contaminated sites. The orange are brownfields. So you can see, there are a lot of brownfields. The red triangles are Superfund sites. And just really I mean, something I noticed from looking at this just quickly, with some, clusters of contaminated sites inland, really the Bay Area is characterized by contaminated sites in the coastal areas, as Rebecca mentioned. And so this is a problem because contaminated sites cause health disparities. And that's because people living near contaminated sites are chronically exposed to hazardous contaminants. And so, I'm sure, with some of your work around, say the impacts of oil refineries and any kind of oil and gas industrial activities near where people live. Where those communities have higher rates of cancers and other health issues as a result of these exposures. And even if the exposures are small, if they're occurring every day over long periods of time, again, these can lead to significant health impacts. And this can happen through kind of innocuous things like dust blowing off these contaminated sites. It can occur through inhaling vapors, and then other forms as well. But in California, and in other places, it's really the dust that can be the biggest way we get exposed to contaminants from contaminated sites. So now I'm showing a figure from the Environmental Protection Agency. So in the green, we're seeing the number of sites, so contaminated sites added to their registry, to that kind of map that you see that we saw before. And you can see that we're adding new sites. So we're essentially, you know, polluting more and more all the time. Um, but the rate of cleanup or remediation is really going down over time. So this, I guess, review ended in 2017. So you can see, in 2017, there was only one contaminated site in the entire country that was cleaned up. And the main reason given for why contaminated site cleanup is slowing down in the US is because of the really high cost of remediation using conventional methods, which we'll talk about. So this prevalence of contaminated sites is a public health and environmental justice issue. And as you know, Rebecca spoke to, and I saw you have an article or like a post, a blog about this. There's a lot of documentation about how, well, a couple about how climate change will impact this. But first, just that actually living within a mile or two of a contaminated site is actually taking years off of our lives. And the amount of years that is taken off of our lives um, gets even worse when there isn't a remediation plan for a site, when the area is also socioeconomically disadvantaged, and when the area is predicted to be impacted by climate change. So it's pretty hard to find an area that isn't predicted to be impacted by climate change. So effectively meaning that, extreme weather, natural disasters, flooding, fires, these can all increase the uh, spread of pollution and exposures to it. So the US Government Accountability Office found in their own review that 60% of the most highly contaminated sites in the US will be affected by natural hazards, flooding, or wildfire. And so I made this simple diagram here because I think we talk often about how pollution leads to climate change, right? So through, greenhouse gas emissions, for example, is one way. But I don't always hear in the discourse people talking about how climate change also leads to pollution by releasing pollution from existing. So say, you think of that. I mean, I've been in LA. The amount of pollution that was created and spread through the fires that just impacted the area is uh, the impact is huge. There's now pollution. I mean, there was already pollution throughout LA, and now there's even more pollution. So I just wanted to really make this link. And then coming back to that same, so this is that same, web tool showing the Bay Area and its contaminated sites. And then I just added a layer showing areas predicted to be impacted by flood hazards and sea level rise. And again, I mean, you can really see that, the Bay Area is predicted to be just significantly impacted by the release of contaminants from all of these sites that are in coastal areas. And a study actually found that there's 423 hazardous sites in coastal California that are predicted to be exposed to a 1 in 100-year flood event in the next century. So now starting to move into, what can we do about it? Just to define remediation, it's the cleanup of this hazardous pollution, these hazardous substances in the environment through their removal, treatment, or containment. And as Rebecca mentioned, and you may know, dig and dump is the most common remediation method because it's fast, like theoretically fast. It involves literally digging up the contaminated soil or sludge or whatever and moving it somewhere else. This costs a lot of money. This is like millions of dollars per site, at least, typically, especially for a highly contaminated, industrial site. And in California, well, not just in California, but yeah, typically contaminated soils are disposed of out of state. So in California specifically, uh, contaminated soils are shipped to Utah, Arizona, or Nevada, and dumped on tribal reserves with hazardous waste landfills there. So this is from the Department of Toxic Substance Control's own report from 2023. The largest waste stream generated in California is actually contaminated soil from these remediation efforts. It's wild because you don't see this, I mean, with the fires, with some of what's happening, we might start to see it more, in the news and the public consciousness, but it's like, I was shocked to learn that this is California's largest waste export, and 56% of contaminated soil from these remediation efforts are shipped out of state for disposal. This releases significant more greenhouse gas emissions than any other remediation method. There's trucks like I always give this example. I was connected to a site in LA. It's 3,000 acres big. They are using dig and dump to dig up all the soil. It is being trucked to Utah. It will take 30 years for that site to have that soil trucked to Utah. Think of how many trucks going back and forth, and it will cost $1 billion to do that one site, which also happens to be a sacred site for all of the tribes of Southern California. So I'm just again trying to, I think my next slide is then linking, or no, I'll get there, but just trying to really make these linkages with climate change, not just in how pollution causes climate change and climate change causes pollution, but also how we remediate can contribute to climate change, or can contribute to climate resilience, which I'll start to get into. So, this question that's been driving me for the last 15 years or so is like, well, what are other remediation options that actually address the pollution, for one, don't just move it somewhere else, that are more environmentally sound and more cost effective? If cost is the major, reason that contaminated sites aren't being cleaned up, and we're facing this like crisis with climate change accelerating the exposures and release of contaminants from these sites, then I was like, there's got to be something else we can do. And fungi are a really important, can play a really important role in any type of remediation, whether it's dealing with hazardous and polluting wastes, or with remediating contaminated sites and waters as well, actually. But so mycoremediation, working with fungi to address pollution, is within this larger category called biological remediation. Bioremediation just refers to working with living organisms to address environmental pollution. So if we add plants in the mix, that's called phytoremediation. Bacteria, it can be called microbial remediation. And so some major advantages here is that it can be done in place. So it can reduce or just completely remove the need for soil to be dug up and dumped out of state. So it's then, preventing those emissions. It's often preferred by, tribes and communities and the public, because you're not, moving dust all around. You're not like destroying land, because the other part of it that I didn't mention yet is when you dig up all that soil and move it somewhere else, you then have to bring in clean soil, which is often digging up and completely destroying or disturbing environments like deserts, and other environments to then, bring in this infill. It's thought to be 50 to 90% cheaper than dig and dump. It can increase biodiversity and repair soil on site, and so many other benefits. So another kind of overly simplified diagram, but just to connect some of these ideas again. So conventional remediation releases greenhouse gas emissions and contributes to pollution. It doesn't address it, it just moves it somewhere else, spreading pollution into other communities. Whereas nature-based remediation or bioremediation, these are in situ methods that actually sink carbon into soil. So they have the opposite effect and so many other benefits, like I mentioned. These nature-based remediation methods can actually help enhance climate resilience and even help mitigate climate change. And having been working on this stuff for a while, I've come up with a few basic principles, things to consider when, considering bioremediation for a site. And so I'll just go through those and start to bring in some case studies as well from my research. So the first one is to design based on contaminants present and site conditions. Second is pick fungi or plants and or bacteria that are already adapted to the contaminants and the site conditions. Third is to mimic nature. And I'll go into that. And then the fourth is to give the fungus or plant what it needs. So for that first one, I just want to cover some basic toxicology. It's relevant to the oil refinery sites as well. So oil refineries, let's just use that example, are going to be full of organic contaminants. Organic contaminants are carbon-based, meaning that they were manufactured or derived from something that was once alive. So, fossil fuels are organic contaminants, petrochemicals, plastics are organic contaminants, technically, because they were made from fossil fuels, which are made from stuff that was once alive and got compressed for a long time, basically, in the earth. So pesticides, I mean, we make like so many things out of oil, essentially. So there's a lot of different types of organic contaminants, but because they're carbon-based, some of them or many of them can be degraded through microbial metabolism, which basically means that, fungi, bacteria can eat them. They apply enzymes to them, and break them down to get the sugar, the food, the carbs that they need for their lives. So oil refineries are going to be full of oil and organic contaminants. They're also likely to have metals. So metals like lead, arsenic, cadmium, chromium, nickel, there's so many. These are examples of inorganic contaminants that can also be called elemental contaminants. They're what you'd find on the periodic table. So the thing about elemental or inorganic contaminants is that most of them cannot be transformed into less toxic forms. And that makes them difficult to remediate because you essentially, I mean, I didn't talk about capping. I personally do not find that to be a wise remediation option because especially under, changing climate conditions, you're basically just covering up the contamination and hoping, usually without monitoring, that it's not being re-released into the water, groundwater, surface water, or, like air or or whatever. So anyways, yeah, metals generally they don't break down, except in a nuclear reactor or like the heart of the sun. And so we typically move them around, or stabilize them, like capping or something. You try to keep, they're still in the soil, but you just try to keep them there. Or you move them out of the soil, either by moving all the soil or by, working with plants and fungi to to pull the metal, to extract the metals from the soil, and then remove them from the site where they can be contained and not cause harm. So this is really important when thinking about a remediation strategy. So sometimes, people will, have lead. Say that you somehow only have lead on in your backyard or something, and you're really excited about fungi. Applying, you know, fungi to the site is probably not going to address the lead. And we'll go into that more as we move through this. So you want to pick species, fungi plants that are already adapted to those contaminants and the site conditions. It's actually a good segue to what I was just talking about. So luckily there are fungi everywhere, and I'm focusing on fungi because they've been really kind of overlooked. There's been more research into, working with plants for remediation. So I'm really focusing on fungi here. But yeah, there have been fungi isolated from plastics floating in the ocean, from electronic waste, from pure oil tailings, from Chernobyl. So, and if you want, I'd encourage you to look at some of these species here. You'll probably see them come up again throughout the presentation. They're not sort of charismatic fungi. These are a lot of molds. Um, Actually, Agaricus bisporus is like the common button mushroom that we eat on pizza. So that I guess is a charismatic mushroom. But the rest of these are pretty much all molds. Some of them toxic molds, like Aspergillus fumigatus is like a black mold. Anyways, the pure oil tailings one is really interesting. This is actually from Canada, where I'm from, and from the tar sands there. Basically, a researcher saw a little, I think it was a dandelion, like a single dandelion growing in literally pure oil tailings. Nothing else was growing. So they collected it and took it back to the lab, and they determined that the reason it anyways that there was a fungus in the roots of this plant, in this dandelion. And then they cultured the fungus, and they did experiments where they planted plants in pure oil tailings. Without the fungus, the plants could not grow. With the fungus, the plants could grow, and also the oil tailings were being broken down by the fungus. So there's fungi everywhere. And so I actually I've used this approach a lot in my work. So we actually went to La Brea tar pits in LA, and it was so cool. We got to literally crawl in the tar pits and like be next to a woolly mammoth skeleton and take samples. And sure enough, we sequenced them and found a bunch of different fungi, but we also cultured them and found black yeasts, black yeast fungi in pure tar. So why are these fungi, growing in pure tar? They're there because there's food for them, right? No organism would stay somewhere where there's no food to fuel their life cycle. So you can usually assume that if there's a plant or fungus or bacteria growing on a contaminated site or pollutant, it's probably able to at least tolerate it. It can at least tolerate those tough conditions and toxic conditions, and it probably is doing something with it, like using it as a food source in some way. So I also took this approach in my PhD in Southern California. I visited 7 Superfund sites in brownfields shown in this map, and I basically just learned about all the plants and fungi and bacteria that were growing naturally on these sites. Not going to go into too much detail here. I'll just point out these are some of the top fungi I found, and you might notice some similarities. Some of the same exact species that were showing up in Chernobyl, in pure oil tailings, in, plastics floating in the ocean. So some of these are fungi that have already been studied for their potential to break down, you know, organic pollutants and interact with metals in some way. Some of them haven't been, but I would guess that that would be a really good, these would be good species to research because they're probably just unknown bioremediators. And I'll just note too, like halophile means it's salt tolerant. Many of these are like not only able to get food from pollution, but they can also do it in really tough conditions, like really salty conditions, or really hot conditions or something like that. Um, I guess I have this slide in here. I'll talk a bit later about one of my favorite groups of fungi, the arbuscular mycorrhizal fungi. Um, you may have heard of them. They, mycorrhizae live in relationships with plants through their roots in beneficial, trade relationships. Um, so the reason I added this slide in, I did also look for these fungi, extracted spores from these sites, and I did find that the most highly contaminated sites had really low, like the quantities of these really important plant helper fungi, and they also had lower diversity. And so the reason I added this slide in, because later we'll talk about different ways you can introduce fungi into a remediation project, and we'll talk about how you may choose to just stimulate and support the fungi that are already present on the site, right? But with some types of fungi that might be really important to the overall kind of ecosystem recovery and pollutant cleanup, they are not going to be stimulated. They're in too low populations. The way they grow isn't gonna be able to really be stimulated in the same way. And so you might consider actually inoculating, like so adding these beneficial fungi back in. Okay, mimic nature and give the fungus or plant what it needs. So just some basic fungal ecology here, again, to help with this selection. Um, so I'm categorizing fungi here based on their ecological role and remediation potential. So decomposer fungi are the ones that we work with most for like an oil refinery remediation, or really when we're dealing with organic contaminants. And that's because they get their food by breaking down dead plants. They break down dead stuff. They eat carbon using enzymes. And again, organic contaminants are carbon-based. So many decomposer fungi have the potential to degrade organic contaminants like petrochemicals through those same enzymes. And they create soil in the process. I'll skip over parasitic fungi for now. Endophytic fungi are really interesting. I also have a job doing plastic degradation with fungi, and there's a really charismatic, kind of well-known, I see it in the news more and more actually lately. There's this endophytic fungus that was isolated from a plant in the Amazon by Yale students, that is like known as the plastic eating fungus. It's an endophyte, which just means that it lives inside of plants, not just in their roots, and in a beneficial relationship. So some of these endophytic fungi can also degrade organic contaminants, or enhance phytoremediation success. And then the mycorrhizae I already mentioned. These are fungi that live in symbiotic relationships with plants through the plant roots. They actually trade elements, like phosphorus, like necessary elements, with plants, in exchange for sugar from the plant. That's how they get their food. And so these same fungi can be applied to enhance phyto removal of metals, which are also elements. And this is like maybe a little out of order. I wanted to um, highlight, since it's not, necessarily a group of mycologists, the distinction between what a mushroom is. So a mushroom is just the reproductive part or fruiting body of certain fungi I'm showing here, some common ones. It's kind of like an apple on an apple tree. It's how the certain fungi reproduce. The actual fungal organism that we're working with in remediation is not like mushrooms, literal mushrooms. It's something called mycelium, which is like the vegetative body of a fungus. It's this web of thread-like structures shown here. It's an actual picture of mycelium. Here's kind of a diagram showing some of those mycorrhizal fungal mycelial networks connected to plant roots. So this living part is what we work with in mycoremediation. And so there's a few uh, particular species of fungi, decomposer fungi, like I mentioned, that have been studied for their capacity to degrade diesel and other organic contaminants from solvents, PAHs, gasoline, and so much more. And I'll go through a few case studies of that later. But one to note that you probably already heard of is the oyster mushroom, or Pleurotus austriatus is its Latin name. And then again, these, if you're dealing with a site that has metals as well, you're probably going to want to have plants in the mix to pull up those metals. And you're probably going to want to have those plants have the support of their mycorrhizal fungal partner. And that's, I've been in this field for a while. It's very rare to not have both metal and organic contaminants. Again, even in oil refinery, there's probably going to be metals in the mix. Metals are often mixed into different uh, refinery processes to help them flow better so that they can get like pumped out, pumped around. So they tend to be present. And so just to kind of pull this model of nature-based remediation all together, and why I've designed it this way is because, yeah, most sites have mixtures of organic and metal contaminants. And in nature, we don't usually see a single type of life taking over, right? We usually see a whole bunch of different plants, a whole bunch of different fungi, a whole bunch of different bacteria. We got worms, we got a whole bunch of stuff all together. And so this model of ecological remediation that I'm working with involves beneficial decomposer fungi that we add to break down those most complex organic contaminants, like petrochemicals or solvents. Um, Typically, you don't even need to add additional, bacteria or additional fungi, because once the decomposer fungi start that breakdown, pre-existing, bacteria and secondary decomposers will now be able to continue the breakdown of stuff that was too complex for them before. And then we have plants, multiple types of plants that are not just any plants that have been identified to hyper accumulate or like pull up huge concentrations of those metals of concern, supported by these mycorrhizal fungi that help them do that better and survive in poor soils. And so it would again be pretty amazing if you were able to achieve a complete remediation with just one species, like just sunflowers or, just oyster mushrooms. It's more likely that having this combination is what will address all the contaminants that are present and help regenerate and restore the whole system and its health. So, there's a few different ways we can we can work to remediate a contaminated site like an oil refinery. And I just want to go through those, and then I'll give a few of those case studies. And so one is natural attenuation. This is something you'll hear certain politicians or lobbyists or whoever industry speak to, where they're like, just let nature do its thing, basically like don't remediate. Nature will heal itself. And to some extent, that's true. It might take, thousands of years and a lot of harm be caused in the meantime. Again, there are probably bacteria and fungi, maybe plants on these sites that are probably there because they're able to metabolize or work with some of that contamination in some way. But what I tend to suggest is some of these other approaches. So bioaugmentation would look like, say, okay, you've seen some studies, you have this oil refinery shutting down. You've seen there's been a few case studies of Pleurotus austratus mushroom effectively degrading, different petrochemicals. It's not going to be, you're not going to find that one growing in the soil naturally on this site. So you decide to introduce it to the site. You introduce a known remediator to play a specific role. That would be bioaugmentation. Biostimulation would be saying, yep, there's definitely fungi and maybe some bacteria. There's already probably some stuff in the soil that will be trying to work on some of these pollutants. I'm going to help them out. I'm going to help them do that faster by giving them usually water, nutrition, like, compost, wood chips, like some easier to digest food to help them kind of grow more and do that faster. And then there's those plant microbial interactions I mentioned, which are especially important for metals. Okay, so then I'm just gonna I have a lot of material here, but I want to do some case studies, and then we can open for questions. So I'm just going to briefly go through my field testing on 3 contaminated sites in LA. There were many, many partners. All of these sites had mixtures of petroleum products, diesel, PAHs, as well as metals like lead and other metals. So I feel like these studies are very relevant to a site like an oil refinery. Really, there's the same mixtures of contaminants and the same types of issues with the soil. Like they can have like high in salt and other issues. One of them was a 100-acre rail yard, actually. And the others were automotive industry related as well. So I actually compared all of those 4 approaches that I just talked about, right? So natural attenuation is basically doing nothing. Those were my controls, right? I didn't do anything. Biostimulation looked like adding wood chips and irrigation and plants to stimulate those mycorrhizal fungi that only grow with plants. I also looked at bioaugmentation. So I spent oyster mushroom blocks from a local farm, and I also got an inoculum of that mycorrhizal fungus, and I added it in. And then I also looked at those plant fungal interactions. So I tested some native plant metal extractors I'd found in that first study I mentioned. I compared it with a known, like kind of superstar non-native metal extractor, and I tested it with all those different combinations of fungi. So what did we learn? Specifically, the Southern California native plants I tested turned out to be pretty effective and like totally underutilized tools for remediation of metals because they're drought adapted. They have really extensive and deep root systems. And those root systems mean that they can reach metals a lot more extensively in the soil. So we did see significant reductions in all of the metals of concern over the 1 year period the study ran. It turned out that adding that mycorrhizal inoculum was the most significant contributor to the metals being pulled out of the soil, and also to plants even being able to survive and grow in these contaminated sites. I probably don't have time to go through this graph, but it's just showing what I just said, that basically without the mycorrhizal fungus, no lead is taken up by the plant. When you have the mycorrhizal fungus, then the plant is able to pull that lead out of the soil, so it can be removed from the site. Surprisingly, the oyster mushroom also increased lead removal by plants, but I don't know why. And excitingly, we did see pretty much a complete removal, so degradation of all the different types of petroleum hydrocarbons when we inoculated with this Pleurotus austriatus fungus. So just to kind of say, like, what I've really learned is that when you mimic nature, so these are before and after pictures of these sites, regeneration happens, remediation happens. It was really astounding to see, like, sites with literally oil soaked, like black soil that you could smell, that I saw catch on fire on hot days, like, be transformed into these kind of beautiful, thriving spaces. So people always want to know, what do you do with the plants that have pulled up the metals? So I've been testing, I collect, you collect all of them. Typically, you will do a controlled incineration or a controlled sledging. So just basically, like, for projects, I pack all the plant biomass into garbage bins without holes and put the lid on, and it anaerobically decomposes over time. And basically, instead of having to remove all of that soil that has the metals and dump it somewhere else, you just pull the metals out of the soil. Now you have a bunch of plant biomass, and then you concentrate that plant biomass into what ends up being a small amount of concentrated metals. And those metals can actually be reused, reducing the need for mining and all of the associated problems with that. So we're basically moving metals from a place they're causing harm and a public health issue and ecosystem health issues, and, extracting them and concentrating them so that they either aren't causing harm anymore or can even be reused potentially. And I've explored other things like making biofuel out of the plant biomass. So I'll just briefly mention that I think I did already, but we're also testing these methods on fire contaminated sites, which have the same mixtures of contaminants that you see on an industrially contaminated site. And, again, this is really trying to link these nature-based remediation methods with climate to kind of mitigate some of the impacts of climate change, like mobilization of pollution. And then, perfect. Okay. So my last couple of slides are specifically about oil refineries. This is not a study that I have carried out. The only case study I found in the world that actually was a field study carried out on a decommissioned oil refinery was in Poland. And they basically cultured fungi that were already growing on the site and then re-inoculated this basically lagoon. And in a 4-year period, this process reduced 81% of the petroleum hydrocarbons, which is, I think, pretty amazing. I mean, they're talking about sulfuric acid based, like, waste. It's highly acidic. It's highly aged, which makes it can be harder to break down. It's covering a huge area. So I thought this was kind of an exciting case study. And it again speaks to that a combination of methods I talked about, right? Where you basically are supporting the fungi that are already present on the site, um, and but amplifying, like helping them by growing more of them and then reintroducing them. There's a couple studies. One of them was done in California. It wasn't um, super successful, but it was a good learning experience. They also use, you'll notice, well, these studies also use that Pleurotus austriatus, that oyster mushroom. This one was done along the, oh my goodness, I'm totally blanking on the name of that river right now. It was done like, anyways, along a river where there had been a diesel spill and impacting the soil and getting into the river. And so they basically inoculated, um, these big, long soil, diesel contaminated soil piles with the oyster mushroom. But they didn't give the fungus what it needed. Um, I didn't talk at length about that, but a lot of times if you see a project, a bioremediation project that didn't work out, it's because the plant or fungus or whatever living organism wasn't given what it needs. And, of course, bioremediation is working with living organisms. So they will not live and do the remediation if they don't have what they need and then they die. There's another study I'm referencing here. There's actually quite a few studies that yeah are not on oil refineries, but are to treat and remediate petroleum hydrocarbon contaminants. So very relevant. Again, this one also used the oyster mushroom. They had pretty good success in a 3 to 4 month period, but they found that they needed to use a huge amount of inoculum for it to work successfully, and that it worked best when combined with compost as well. So the last thing I'll mention is that we need to build a workforce that has these skills to apply these projects on a larger scale, such as something like an oil refinery. Is it going to be a big project? So I've been working on a lot of workforce development and trainings with youth. So there's a high school program we run in LA School District. We're actually talking with Oakland United School District about and some other projects up in the Bay Area about running a similar program. But it's just a mix of theory and hands-on learning that basically connects high school students with a bioremediation pilot project, which they help with and learn. And they get their HAZWOPER certification, which is what you need to work in this field. We've also run it with a lot of other community groups. And so really what I'm proposing here is a much more sustainable remediation model, right? You don't have those emissions from shipping soil. It regenerates soils so that it can then support plants and trees, which of course can help sink carbon along with the fungi. We're cleaning up contaminated sites because this is so much cheaper. It means that a lot more contaminated sites could be cleaned up, right? Which would reduce risks to fence line communities. Again, yeah, these sites can often be transformed into parks and green spaces or community gardens, which have all their own benefits. Of course, like clean soil is essential to clean water. And so if we don't clean up these contaminated sites, our water is also impacted. And again, we can build a regenerative economy and workforce with local jobs with some of these approaches as well. So with that, I'll end my presentation and share some of my contact information. Um, before I open for questions, I always forget to mention, like, yeah, I do run a nonprofit based in California. We work all over, but um, we are implementing these projects uh, in collaboration with community partners. We're doing these trainings and workforce programs as well. So yeah, and we do have a growing number in the Bay Area. So if this is something you're interested in, please don't hesitate to reach out or check out our website. We have a kind of like an interest form for collaboration or remediation projects and trainings and things like that. So.
Thank you so much. I mean, this is just information packed, to say the least. You've gone to an awful lot of effort to put this together. I see we have a question from Shoshana. Shoshana, go ahead.
Let me unmask. Danielle, I am completely blown out. You have just descended from the heavens. Thank you so much. And I want to thank also Becca. Thank you for facilitating, for putting together this opportunity for us to experience you, Danielle, and your amazing information. And also Jean Wu, who happened to be in Santa Cruz, who heard you speak and brought her excitement to this group. I want to say that I'm also affiliated with the Sunflower Alliance. The name speaks for itself. We have been involved in basically, blocking the expansion of hideous fossil fuel projects for the last few years. But ultimately, we took the name from a plant that is known for its, hydrocarbon remediation, and ultimately it's about regeneration and remediation. So I want to kind of introduce an idea here, and I hope Maureen Brennan will follow me. We have been in contact with Phillips 66, which converted to biorefining, not such a groovy thing, really. Um, and in the course of that conversion, um, decommissioned its carbon plant, which is a calcining plant, which produces pet coke. And so we're looking at 20 acres of really uber contaminated soil. And sadly, P66 basically wants to kind of turn its lease over to another polluter. The Contra Costa County is all good with it. And we have actually already met with both the county and with the PR guy at P66, who kind of was tickled by the idea of maybe turning over these 20 acres to a research project.
Oh.
So we've already kind of, and I hope Maureen, who is really integral to this, will follow me and describe a little more about what's going on. I had no idea that you existed, but I think this is perfect for you. So I just want to say that let's talk offline.
Oh, yes.
Let's make this happen. This is just so brilliant. Yes, yes, yes.
Thank you
Thank
so much,
you.
Shoshana, for sharing that. I would love to follow up.
Fantastic.
Love to.
Yeah.
Maureen.
Hello, there we go. I will follow up. And thank you. This information is fascinating to me. I've been a microbiologist for 50 years. And I mean, in the clinical lab setting, but doggone it, these fungus can do the job. I've seen it. I saw it up in Prince William Sound.
Yep.
When there was oil all over the beaches. I mean, 40 years ago, but et cetera. But we have two really important projects going on where I live in Contra Costa County, and one of them Shoshana described. They're decommissioning a carbon plant, Phillips 66. On top of that, they've also decommissioned Santa Maria. And these sites are quite contaminated, And but for the carbon plant, they have $30 million in
Yeah.
their work plan to clean it up. And what does that mean? I'm not sure, but I really wish that your group or a scribe person can help us with this. This would be a perfect place to test out this possibility. And the other area nearby myself is Selby Slag Site, and it's fascinating because it's heavy metals. It was an old smelter site. These are all within 10 miles of where I live. And there are these toxic sites they're all over the Bay Area, but this is so fascinating. And I'd love to, with Shoshana, talk to you offsite. I mean, offline.
Oh, I love that. Thank you. Let's definitely follow up.
This is inspiring. Thank you.
Oh, thank you.
Shirley.
Good. Thank you, Rebecca. And thank you, Dr. Stevenson. This is really amazing. But I'm just looking at, can you tell me how deep, or does it vary, the depth of the toxic material or soil? And how deep do these roots or these plants able to mitigate that and pull it? Because you have the before and after, and the green part looks great, but is there further depth in that hasn't been reached, is my question. So
Great question. So, yeah, so typical science answer, but it really depends. So without doing any type of digging or using machinery or anything, just with simply planting or inoculating directly on the surface of the soil and letting, that mycelium and plant roots penetrate down. In Southern California, so dry, conditions kind of hemmed in by summer, like really hot season, hot, dry season, you can get about, the average is 2.5 feet per year. And so that's actually typically can be suitable, because a lot of times the highest contamination is actually in the surface soil in the top few feet. That's not always the case. Some sites are going to have contamination that's much deeper, and in those cases we either work with species that can go deeper. Some of the species we work with have roots or mycelium that can go like 15 feet, 30 feet in a year or so. Or in some cases, based on need, we will, work with machinery. So I've done that with so at that rail yard site, for example, we had a treatment area where we did essentially make trenches and then inoculated. So we were able to address the contamination that was again, without doing that, the fungi we were working with there could grow on their own, like about 3-4 feet down. But then you start to get oxygen limitations. So yeah, so basically, yeah, there's a lot of things to consider, but we always design the strategy to address the depths and types of contaminants present. And the goal is always to get them to below the screening levels for, regulatory agencies to consider the site clean and safe.
That's great. Thank you. It's fascinating. I hope you get some kind of Nobel Prize for this.
Oh, thank you.
Susan.
Hi there, Dr. Stevenson. Great presentation. Thank you. I had the privilege in my professional career of working with several environmental engineering firms, and one of them in particular was, I think, pretty far ahead in their bioremediation approaches. And I was wondering what kind of affiliation you have with firms that are working in that space. By the way, I also have another question, but let's finish this one first. Do you work at all with firms who are working in that space?
Oh, for sure. I work with engineering firms on most projects. So, because often an engineering firm, so I've worked with like AECOM, Tetra Tech, Jacobs, what are they? Oh my goodness.
Yeah,
I'm
those are the
worked with
McGillas, the big guys.
Yeah, I've worked with a lot of the big guys, but I've also worked with smaller engineering firms. Yeah, so typically we are required to have our work plans or some level of oversight from a professional engineer. That's just required by DTSC.
Yeah.
Yeah, and often, they will have gotten, say, the contract,
Right.
for some, whatever, phase one, phase two, or even maybe the remediation, and they'll like subcontract or partner with our, center or something to carry out a bioremediation pilot or something like that. So yeah, definitely work a ton with engineers. I'm trying to hire. We have also professional environmental engineers that we're bringing onto our team as well.
So maybe I'll just send you an email about this particular firm to make sure you know about them, because it might be a beneficial collaboration. So that was first thing. The second is kind of a general question. I came away appreciating maybe how unique each site is, and how the solution might really need to be different for it, depending on not only the contaminants but the conditions. And so my question is, when you go after a particular site, is there a period of kind of testing and experimentation and piloting and before you go to scale to work on the site as a whole? And if so, how long does that period take?
Very astute observation. Yes, absolutely. With biological approaches, each site is unique. I tend to work with a regional approach, so I will kind of so to answer kind of your second question, I will do that. Well, we always thoroughly characterize every site that we'll be working on, and that can take a few months. It can take a few days. It totally depends on like the size of the site and other factors. But yeah, we do. If there hasn't been like phase one and phase two site assessments, which is like an official EPA thing,
Right.
even if there has been, we typically do additional testing. We do comprehensive soil testing. We analyze the soil. We analyze the site conditions. So that's done for every site, and then we'll tailor a specific design, essentially remediation design, to the site conditions. The kind of palette of species we'll pick from to plug kind of add into that design for a specific site tends to come from a regional focused assessment that only needs to be carried out once typically. So like when I first moved to Southern California, I did that study. I visited those 7 contaminated sites, identified plant and fungal and bacterial, communities that are growing on those sites, figured out which ones were doing remediation and work. So do you see what I'm saying? So like,
Yeah.
that kind of is where you get your kind of regional palette, I guess, of species to work with. And then you assess and characterize each site and put together a work plan that'll address the contaminants and depths and be tailored to the site conditions.
So in a place like the Bay Area where we have so many,
Yeah.
And where possibly a lot of these refineries will be shutting down in a relatively short period of time, over a couple or 3 years or maybe 4, I don't know. But having done the regional piece,
Yeah.
might make the specific site characterizations shorter. So
Exactly.
it doesn't have to be, it doesn't have to take forever to start the
Yeah,
process of
and
remediation. That was really my question. Yeah.
absolutely. And luckily, and that's, I was so excited to be invited to speak to your club, because I already have projects starting in West Oakland, and in
Oh,
for other parts of the Bay Area. So
nice.
we're already starting
Oh,
that
good.
kind of first regional, like kind of screening or kind of initial testing. So yeah, it should be then just that.
That's great to hear. Thank you so much.
Thank you.
Claire.
Hi. So I just was sort of interested if you could clarify. I think you mentioned during your talk that in one of the pilot sites you had an 81% decrease in the particular target contaminant over 3 years. But then you mentioned during our discussion that bioremediation is able to get soil to the point of meeting EPA standards or whatever is considered a cleaned up site. So I was sort of wondering how to crosswalk between, an 81% decrease, is that for those 3 years and then so on and so on. And also, are you able, does that get you to a clean site according to the standards? And does that require having, multiple interventions targeted to the different contaminants at a site?
That's a great question. So the 81% specifically was from that Polish study at the oil refinery. So it wasn't my own study. And they just happened to report in a percentage reduction. It can be common to do that because it's a little bit easier to kind of comprehend, like, uh, but you're missing a really important part, which, like, you're bringing up, like, well, what did it start at and what did it end up at, and where is that above or below the the regulatory screening limit. So but those screening limits are really different, even state by state, country by country, agency by agency. So I think that's why sometimes people report it as like a percentage instead. Anyway, so that one wasn't my study. I did also report some percentage reductions just for this presentation. In my scientific publications, I have like typically how far they are from being considered clean, or and then graphs showing like the line with the screening limit for that contaminant, and then how where it was before, where it was after. So yeah, for my own studies, I mean, it depends on the contaminant, which is also why it's hard to represent if you have so many different contaminants and you're trying to show them with all the different levels. But in my own studies, I've had really good success in a short time period, like 3 to 12 months, of reducing the organic contaminants to completely below, like literally pretty much gone, but definitely well below the screening limits. The metals can be trickier. And by trickier, I just mean they can just take longer. So that's where, like you suggest, you may need either just more time or multiple rounds of planting, multiple, treatment avenues. So yeah, metals, especially lead. Lead is difficult to remove from soil with plants, and that's why I was so excited by that finding, that actually adding this particular mycorrhizal fungus really makes all the difference in helping plants remove lead from soil. So yeah, so with my own studies with lead, we did not, one of those three sites did actually get below the screening limits. But, as has been mentioned, I did 3 sites. They were so different. They were different sizes, different concentrations of contaminants, different exact mixtures of contaminants, and so different soil types, textures, everything. So, I found that interesting that with the same treatment and a regional, across totally different sites, we still were able to achieve some remediation, whether it went all the way below screening limits for every contaminant. Um, you know, it didn't end up going that way for lead specifically, although it did for the organic contaminants. Sorry, it's very nuanced, but it's a really good question. Ultimately, and those were studies when we're doing a remediation like pilot or just a contract. We always we try to estimate how long it will take, give ourselves as much time as possible, and then we basically just keep going until we get it below those screening limits.
Susan, did you have an additional question, or should I take your hand down?
No, it's me, Susan, right?
Okay.
I did have a
Maureen,
question.
Maureen, why don't you go?
Thank you for letting me go again. Because I'm wearing a different hat right now. And yeah, I'm on the board for the Rodeo Sanitation District in the Bay Area. And as with any sanitation district, we have a problem with nutrient load like phosphorus and nitrogen that gets outflowed to the bay. We're very small, but there's like huge facilities. We also have problems with pharmaceuticals that can't filter those out, PFAS issue.
PFAS. Yep.
And can fungus bacteria models be applied to these issues for a sanitation district?
Definitely. Yeah, I've actually done it's been a little while. I have a few projects right now that are focused on water, but I started out doing quite a lot of work around different treatment facilities, stormwater filtration, kind of riparian filtration. Now I'm working on I've done a little bit of groundwater, and so yeah, lots of different types of water cleanup with fungi are possible. I have worked with fungi to degrade PFAS successfully, and definitely, I mean, it's extremely effective to work with fungi to address those nitrates and phosphates and stuff, as well as some of those pharmaceutical compounds. Yeah, definitely feasible. It would probably start with a pilot test, like a small scale, especially working with depending if it's what the scale is, municipal or smaller, but it's always good to start with like a small scale test, kind of show people its efficacy, and then kind of apply a small pilot and go from there.
Oh, that's wonderful. I mean, we are a very small district. We're called micro district. We would be the perfect test place.
Awesome,
Oh, we
cool.
must talk.
That's so fun. I was really wanting to get to do more water stuff again. So that's great. Because
Lovely.
again, yeah, I grew up, I started by saying, I grew up on polluted Lake Erie. And so I really kind of got into this from that concern around water pollution.
Around
And then,
water.
so yeah.
Oh, thank you.
Susan, did you have a question?
I was just curious, like you started with like the vastness of the problem, and how much contamination there is. And I just wondered like what your sense is of like how much of a fix this really could be. Is this like, I mean, is this a way that we could fix most of this I mean, assuming that we put in a lot of effort to do that, or is this like, there's a certain percentage of these places that would be able to be remediated this way?
Thanks for that question. I don't think I've been asked that before, and I really appreciate that. Yeah, so this is a really scalable method. Again, if we have a trained workforce, if we have folks that can help out, and we, can get the materials together. The materials needed for this are very accessible and very cheap. Seeds, and many of them can be collected for free, like, potentially seeds, spent blocks from a mushroom farm, potentially, or like wood chips from the city, things like that. But I think there's a huge potential for these methods to be applied if we get some of those resources in place, on a pretty large scale. Currently, I do not think that the method or the technology is able to deal with, like, for example, I've never worked on radioactive pollution. So if there's a site that has radioactive contamination, I think there's potential. We're not there yet. So yeah, maybe one day, but I think there's some sites that are just so highly polluted. And I don't even mean an oil refinery, but I do mean maybe like again, like a nuclear testing range or something. Like, it's just so highly polluted and with just really intense contaminants that, though probably the safest thing for the environment and people would be to just really effectively contain those, in the meantime. So that's kind of my sense. Yeah, huge potential probably for huge swaths of land, everything from like former mining lands, oil refineries, brownfields, Superfund sites, all kinds of sites, but maybe, yeah, like nuclear testing ranges or things like that. Not right now. We can get there though. Like, people said PFAS wasn't able to be biodegraded, and now, we know fungi can break it down. So it's, we might get there if we devote resources to it.
Danielle, this is Jean. Thank you so much.
Hi.
Yeah, hi.
Thank you.
Yeah, I just was really, it's just amazing to me what you've been able to do. And then we look at these oil refineries going away, and this is an opportunity. This is a really great place to do this. I'm curious, what fungi work with PFAS?
Some of the same fungi. I mean, so you'll notice, like, Pleurotus austriatus came up a lot on this list, or in these different studies. A lot of common fungal molds, like, and yeast, like penicillium, literally,
Really?
aspergillus, yeah, you really tend to see the same ones. And then I'm thinking for PFAS specifically, I think there's even some additional ones that I might not be aware of, that also work on PFAS.
That's great. I did have one more question about how low the lead levels went when you remediated. Yeah. How far down did you get those levels?
Like on average per site? Yeah. Let me think back. So I could pull it up. So, well, okay, first let me say, so for those 3 sites, one of them had lead as high as like 25,000 parts per million, which is insane. One of them was like in the middle, is like 1,000 parts per million or so. And then one site had lower levels. They're still elevated, but they were like between like 300 and 500 or so. The site that had the lowest levels, that's the site that ended up being clean. Like, the lead was reduced by like below the screening limit of 80.
Oh, you went down to 80. Okay, that's what I was curious
Or
about.
below, yeah, 80 or below. The other sites, it was just really variable. And it was so variable that it changes the average a lot. But that makes sense. So it's, yeah. So we had really significant lead reductions. It's just that they didn't get all the way down in the 12-month period. It did seem that you know, if there had been more time, we might have been able to get there.
What's interesting about Contra Costa is there are these hidden or lost smelter sites that have never actually been clearly identified. And it would really be interesting to see how well you're able to get the lead down on a smelter site, because I think there are numerous sites like that. Also salvage yards, there are old salvage yards that need to be remediated, but no one actually knows where they are. So it's
Exactly.
yeah, Richmond is, you know, full of that stuff. So yeah, no, thank you. I'm really hoping to work with you on some of these when you climb up, and it would just be fun to do. Yeah, that'd be great.
Oh, me too. Thank you so much, Jean, for like making this connection. And oh, there was two quick things I wanted to say, but I forget what. Oh yeah, the first one is, I mean, the funny thing about the time part is that, like I said, like dig and dump is theoretically faster, but it's obviously not actually faster in practice, because the entire country is full of contaminated sites that aren't being cleaned up. So like, because it's so expensive and there isn't space to dump all of the entire country of soil, the whole 65% of the world. We can't just move that somewhere. So like some of these methods that take longer potentially just, I think, makes so much sense. It's like I'm trying to just let's put the pressure on to get access to some of these sites, and obviously with safety protection for the workers, for us, like just start the planting and inoculating, and so that, I mean, there's so many other benefits. Then if there's flooding, there's the soil is held in place, the contaminants are held, you know, like there's just so many reasons that it'd be great to do kind of a massive
once it becomes something that, as you were saying, working with the engineering firms and making it a part of the standard practice, there's a serious injustice about digging up contaminated land and then dumping it on Indian land. That is such an incredible injustice. It should not be happening. So great.
The last thing I was going to say is that I work with the Center for Creative Land Recycling. They're a great resource. They help communities get funding for remediation efforts, basically. And we actually potentially have some a source of funding for Bay Area bioremediation project. So we're just kind of evaluating a bunch of possible sites right now, but the criteria are the funder wants it to be beneficial to a community that's been impacted or burdened by the pollution. So I just wanted to mention that in case any of these potential sites that have been mentioned could fit. There might be a timeline piece. I know the oil refinery is not shutting down till like next spring or something, but yeah, just to put it out there in case one of these sites might be a match.
Yes, I think that Rodeo probably is ready to go. And Jean, I just wanted to add, you know, it's not just indigenous communities that are impacted, but for example, I mean, my impression of standard practice with DTSC and Danielle, correct me if I'm like exaggerating here, but you know, it's very much about digging and trucking. And when I first was exposed to all of this, it was via the AstraZeneca site proposal, DTSC's proposal in Richmond. And basically, it was, you know, we're going to remove soil, we're going to take it down to Kettleman City, which is an environmental justice community, I think kind of at the northwestern edge of Kern County, which is so hideously polluted.
It's horrible. Yeah.
It's horrible. And it was going to be dumped on Kettleman. So from one EJ community to another. Plus the
birth defects
and the diesel pollution.
There's so many birth defects in Kettleman City.
Yeah.
It's absolutely horrible. Yeah, no, this is definitely a really positive move to get away from doing that kind of dumping.
Right.
And you know, creating another disaster somewhere else. Absolutely. Yeah, I forgot about AstraZeneca. That was one of the
And this
That's
business
enrichment.
of, capping sites and then, building or proposing the building of low income housing where you can't plant gardens, which we will need going forward, where
Yeah.
children can't play in the contaminated soil.
Correct. Right.
Oh my God. You know what, as if we're not already living in a dystopia, let's just, you know, just keep digging in further and further and this.
Well, I am so grateful that Danielle was able to give this talk, and everyone here
Yeah.
has so many great projects to work on. Danielle, I need to talk to you also. I found out that there is a park in Berkeley that they want to remediate, and I don't know enough about what's wrong with it. So I have to find out more. It's called Strawberry Park, and it's used to be a railroad yard. So there's probably a lot of contaminants in the soil.
Oh, that's so interesting. I just met with I think it's called Alston Community Garden group about another site in Berkeley that they're trying to start a community garden and it's polluted. I forget what it was like a I don't know. Anyways, and they were asking me, they're like, do you know about the strawberry? What is this site called?
I think it's called Strawberry Park, but it may be Strawberry Canyon Park.
Yeah, they were like, do you know about that site? Because that's also a and I was like, no, I haven't heard of it. But
no, that would be perfect. Yeah,
cool.
perfectly. Yeah, the two of them. Yeah, and TCE, is that something that fungi can eat?
Oh yes. Yeah, I'm working on a former laundromat currently in Torrance. That's all TCE and PCE. And actually there was TCE and PCE on that rail yard on one of the previous sites I worked on too. Yep.
Oh, that's fantastic. Well, I'm gonna stop speaking.
Okay, so we need to let our guest go. I have to say, though, I have two questions to finish up.
Sure.
You mentioned that you're active in West Oakland. Could you just say a few words about what you're doing there?
Yeah, so this is a former auto wreckers shop, and it's now been, I guess, like leased, I think, by a community organization called Neighborship. They want to build low income like tiny homes essentially. And they engaged with me. They got a grant to do the remediation, and I'm working with them on doing a bioremediation project with that workforce component. So we're trying to hire the income, the whole neighborhood is very low income, and we're trying to hire people from the neighborhood to help with the project and get trained. It hasn't started yet. We've had a lot of delays in DTSCs, but they just, yeah. So we're raring to go. We got an extension. So we're just waiting to get approval and then hoping that we can get started planting and inoculating maybe later this summer or fall.
Wow.
Mm. And one last question. You'd mentioned Oakland Unified School District. Could you say a few more words about that? Because we have an active program to engage high schools to encourage climate literacy. And Oakland Unified was actually our first district. So it would be interesting to see if we could extend what you're doing at Oakland, perhaps into other districts.
Oh, definitely. And actually, that's how like the high school program started in partnership with like yourself, another organization that already was doing a sort of climate and environmental after school program in high schools in LA. And then we kind of added this as a additional part of that program. But yeah, I haven't heard too much back. But basically, there's a contaminated site that is owned by the school district, and they were evaluating how to deal with it. And then I think was it Jean again? Maybe Jean connected us, but someone somehow connected us and said, well, hey, here's an option for remediation, and then two, and I know it could be sort of controversial to think about like involving or educating youth in dealing with toxic stuff. But I mean, I will say, like, I have heard from, I mean, I wish I had this education when I was in school. We were told like the world is ending, but you can change your light bulbs. And it just was like, well, what? Like, so I have heard from just about every student that's been through the program that like they feel a sense of hope. They were transformed from feeling like they could actually do something beneficial in their neighborhood to help the environment. And many of them go on to, from not even maybe wanting to go to further school or anything, to be like, I want to do this for my job. I want to study this. So, yeah, so I haven't heard much back other than they were kind of, I told them, about the high school program and then the possibility of just partnering on a remediation project. And they were kind of like seeing if that might fit for them last I heard. Yeah.
Okay. All right. So to finish up, you have obviously an awful lot of interested folks here who are going to want to engage with you. What is the best way for people to reach out to you?
Email is a really good way. So I'll put my email in the chat. Well, let me put
I took the liberty of putting your contact link in the chat and also the main page of your intake form. If
Oh
people
my
want to use that.
Thank
So,
you, Tom.
yeah, sure.
That's
You're welcome.
awesome.
I'm super impressed by your work and the results. It's very encouraging.
Thank you.
Thank you, Tom.
All
What
right.
a wonderful group. It's so nice. I have to say, it's just really nice for me to give a talk to such an engaged group that are all, taking action in your community on the environment. It's really special. I'm really excited to hopefully, get to work more with some of you. So thank you for the invitation. Thanks, Jean, for connecting us, and thanks, Rebecca, for organizing this.
You're very welcome. Thank you for all your efforts.
Thanks, George.
You're here.
All right.
All right.
Okay.
Have a good
Take
evening.
Thank you.
Thanks, Doctor.
Thank you.
Bye.
Thanks, Rebecca.
Thank you, Shirley.
Great meeting. Great
To
meeting.
be continued.
Yes, to be continued.
I'm nominating her for some Nobel Prize, I swear.
You're here.
Well, if Trump can get nominated, then
No, no.
We didn't hear that. No. Oh.
all right.
Cheers.
Take care.
Thank you. Have a good night. Thank you, Jean, very much for putting this together.